everyone, so this is Ashley and I'm the TA for Psychology 1011 this year. Last Thursday on October the 30th I gave a review lecture and a practice test for your exam 2, and a lot of students were concerned that the materials I were covering were not the things that you had covered in your classes. So once I checked into it, I found that you guys were in fact correct about that. So what happened was, while you are responsible for most of the materials that I covered in that lecture, the materials that I was focusing on the most were things that were from your textbook. So it's actually not something that you would have to focus on the most, as the majority of the questions on your exam will be directly from your lectures. So after talking to Dr. Stevens, we decided that uploading a video of me doing a new review lecture might be helpful for you guys. So this is the new lecture, and it's going to be more tailored towards what you guys learned in class. So the first thing I have here for lecture 1 is sensation versus perception. So sensation involves the detection of sensory information, whereas perception involves making sense of the sensory information and giving it meaning. And so there's some more terms and descriptions on the slide, so you can pause it if you want to take a look and learn a little bit about these two terms. So here I have some examples of the two. So for example, sensation might be sensing the warmth of a candle, whereas perception might be perceiving that the candle is near you from that warmth. Another example could be sensing sound that's coming in versus perceiving music, so a particular constellation of sounds together. So now I'm going to go over a couple of examples of absolute threshold, discrimination threshold, and Weber's law. So absolute threshold is how intense a stimulus has to be in order for it to be detected half of the time. So if I was doing a hearing test and a 10 decibel tone was played to me, and on 50% of the trials I said I hear a tone, and on 50% of the trials I was not sensitive enough to hear the 10 decibel tone, we would say that my absolute threshold was in fact then 10 decibels. Now, a discrimination threshold, also called the just noticeable difference, refers to how much a stimulus has to change in order for me to detect that it has changed. So in a hearing test, if I was listening to a pitch at 3000 hertz and then I heard a series of pitches at increasing, a series of tones at increasing pitch, so say 3100 hertz, 3200 hertz, etc. If the first sound that I could detect as being that's different, so okay, this sound is different from the one that I originally heard, so if that occurred at 3500 hertz, then my just noticeable difference for detecting pitch would be 500 hertz. So it would be the difference between the original tone that I heard and the very first tone coming after that I could actually identify as being different from the original. So the next term I have here is Weber's law. So this just states that the just noticeable difference that we just talked about is proportional to the magnitude of the stimulus. So the smaller something is, the less of a change is needed to be detected, and vice versa. So now I'm just going to run over signal detection theory very quickly. So if a signal is present and the respondent suggests that yes, it is present, that is referred to as a hit. So for example, in my hearing test, if the pitch of a sound had changed and I said yes, the pitch has changed, then that's a hit. Now if the signal is present but I said that it wasn't there, that would be a miss. So in my hearing test, if the pitch of a sound did change but I said that it didn't, that would be a miss. Now the next term I'm going to go over is a false alarm. So this occurs when the signal is absent, but the respondent says that it is there. So in my hearing test, if a pitch did not change, but I said it did, this was a false alarm. And the last term I'm going to go over is a correct rejection. So this happens when the signal is absent, and the respondent says that yes, there actually is no signal happening here. So in my hearing test, if the pitch of the tone did not change, and I said that it hadn't changed, then that would be a correct rejection. So I was right in saying that there was no change there, or there was no signal present. So just like in my Thursday lecture, I have a list of terms here for you. An important note for this particular lecture is that on all of the term slides, those that are on the left side came directly from your lecture, whereas those that came from the right side came from your textbook. So they were within your required reading, so they are fair game for a question or two on the exam, but they were not the main focus of the lectures that you had. So now I'm going to go into vision. So as you can see here, I have a diagram of the eye, and I'm just going to quickly go through the different parts of the eye and how vision passes through them. So the very first thing we have is the cornea, So this is a hard, clear shield that protects the pupil. And this is the first structure that light entering the eye will pass through. So following the cornea, light entering the eye will then pass through the pupil. This is a hole that's in front of the eye and it leads to its interior. So the iris is next, and this is a colored part of the eye. And while light doesn't directly pass through the structure, I put it up here because it is still important in vision, because it adjusts the size of the pupil in order to control the amount of light that enters the eye. So once the light passes through the pupil, it hits the lens. So the lens changes shape in order to focus the light through it onto the retina. Now inside the eye, we have the vitreous gel. So this is what maintains the shape of the eye, and light passes through it on its way from the lens to the retina. So here at the back of the eye, we have the retina and the fovea. The retina is a thin layer of nerve tissue that lines the inside of the eye. This is where the rods are, the cones, the ganglion cells, and the bipolar cells, and it's where light is processed first. So the fovea is an area of the retina that has the highest concentration of cones, and it actually has no rods. There's no rods in the fovea. And we see images with the greatest visual acuity when they are focused onto the fovea. So we see images with the best clarity when the light hits this particular area. Now once the light is processed by the cells in the retina, it's sent onto the optic nerve, which carries the information to the brain. Now the area where the optic nerve connects with the inside of the eye has no rods or cones. This is called the optic disc, and it actually creates your blind spot. So the next thing I have here for you is a cross-section of the actual retina. So you can see back in the back, facing away from your eye, are your photoreceptors. So those are your rods and your cones. And then the next level we have coming towards the front of the eye are the bipolar cells, and then finally the ganglion cells. And now there's two other important types of cells listed here as well, the horizontal cells and amacrine cells. Now what the horizontal cells do is they actually connect the bipolar cells laterally, and then the ganglion cells are connected laterally by these amacrine cells. Now when light passes into the eye, it actually goes through all these layers of cells, so it hits the ganglion cells, then the bipolar cells, then the rods and cones, before it actually has an effect in terms of transduction. So that actually occurs first in the rods and the cones, and then the neural impulse is sent backwards through these layers up to the ganglion cells. And it's the ganglion cells that synapse with the optic nerve and actually help carry that information to the optic nerve, and then from the optic nerve to the rest of the brain. So the next thing I have here is a comparison between rods and cones for you. So rods have the highest density outside of the fovea, whereas cones are located inside and outside of the fovea, but the highest density of them is in the fovea. There are no rods in the fovea at all. There's 120 million rods, approximately, and only approximately 6 million cones. Rods are most sensitive to light, so that's what helps you see in dim lit areas or in the dark. And cones are used in color vision, and they actually need quite a lot of light in order to work. Now the next thing I'm going to mention is something that we did talk about on Thursday, but again, it's one of those things that is in your book, so you are responsible for it for a question or two on the test, but it's not the focus of your lecture, and that is that rods contain rhodopsin, whereas cones contain photoopsins, and there are actually 3 types of those. So again, here are some terms for you, and as with the last term slide and the rest for this lecture, everything on the left came right from your slides, and then everything from the right side is actually some extra information that came from your textbook. So still fair game for a question or two, but not your focus. So now I'm going to go over audition. So again, this is hearing. So it starts with the pinna, which is the actual outside of your ear, and that's on the outside of your head, and this directs sound waves into your auditory canal, which brings them to your tympanic membrane, which is your eardrum. Now the tympanic membrane vibrates up against the 3 ossicles, or 3 bones, of the inner ear. So those are the hammer, anvil, and stirrup, and those vibrate in succession. Finally, the stirrup vibrates and presses up against the oval window, which is another membrane that is the beginning of the inner ear. Now the vibrations continue to the cochlea, and we'll talk about what happens there in a minute in more detail, but this is where transduction occurs, and then that information is sent on to the auditory nerve. So here's a cross section of the cochlea. So on the bottom here we can see we have the basilar membrane. Then we have the hair cells of the basilar membrane with some cilia attached to them. Those are like the little hair fibers. And then on the very top we have the tectorial membrane. So what happens is as those vibrations come through the cochlea, the basilar membrane actually moves back and forth and up and down because it's very flexible, so those vibrations can move it. And of course, because the hair cells are attached to it, those move as well. But now the tectorial membrane is not flexible, it's quite rigid. So when those hair cells move up against it, they actually bend and press back and forth in different directions. So if you think about taking a hairbrush and pressing it up against a desk or a table, the little bristles of the hairbrush would bend depending on where you're pressing it, and that's also that's sort of a good analogy for what's happening here with the little cilia of these hair cells when they press up against the tectorial membrane. Now it's this movement of the cilia against the tectorial membrane that actually cause the hair cells to fire. So it causes them to fire an action potential, and then that information is transmitted onto the axons of the auditory nerve. So this is how transduction occurs within the cochlea. So the next thing I have here is just an explanation of how we code for sound in the cochlea. So I'm going to talk about loudness and pitch. So loudness again refers to the amplitude or the intensity of a sound, so it's measured in decibels, and it's coded in two ways. One is by the degree of hair cell bending, so how much are they bending, and the other one is that there are different thresholds for firing for the different kinds of hair cells that are in that are attached to the basilar membrane. So louder sounds will stimulate the neurons with the higher thresholds to actually fire. So now I'm going to talk about pitch. So this is about the frequency, the frequency determines pitch, and it's measured in hertz. So there are two theories as to how pitch is coded within the cochlea. One is frequency theory. So this occurs when the pitch is 3,000 hertz or lower. And these pitches are coded because the frequency of the nerve impulses that are sent to the brain, they actually match the frequency of the sound itself. So they match the frequency of the wave. Now the next one is place theory. So this suggests that pitch is coded by stimulation at different places along the basilar membrane. So high pitches are coded near the oval window, and low pitches are coded further into the cochlea. So here are some terms for lecture 3, and you can pause it here if you like and test yourself with them. So now I'm going to move on to lecture 4, which is taste, smell, and touch. So taste and smell both work in similar ways. The receptors involved with taste and smell respond to chemical molecules rather than other forms of energy, so rather than physical, like in hearing. Olfactory sensory neurons, those are our scent receptors, and they are long cells that actually descend from the brain through our cribriform plate, which is the bone at the top of our nasal cavity, and right into the mucous membrane inside the nasal cavity. You can sort of see this on the diagram I have here. So the sensory neurons that are extending down there have cilia, and they convert chemical information from odor molecules into the neural impulses. So that's how transduction occurs. When the odor molecules come into contact with the receptors on the cilia, transduction occurs and the olfactory information travels to the olfactory bulb in the forebrain. And you can see the olfactory bulb on the little image to the right here. And then this structure sends the information either directly to the primary olfactory cortex, which you can also see there, or indirectly through the thalamus. Now, how we determine different types of smells is that specific constellations of odor receptor activation at particular quantities gives us these different types of smells. And there are as many as 1,000 different olfactory sensory receptors. So here I have a diagram of a taste bud. So bumps on the tongue are called papillae, and these contain about 10,000 taste buds. There are multiple taste receptors inside the taste buds, and this is what actually processes taste information. So it's not the taste bud itself, but the actual taste cells that are within it. So different tastes use different mechanisms to stimulate an impulse in the taste cell, and these signals then travel down fibers to the brainstem and then onto the thalamus. So now I'm going to move on to talk about touch. So we sense touch from mechanoreceptors. So these are receptor cells in the top layer of our skin, and they can be specialized to detect different types of qualities, so shape, grooves, and vibrations, just as a couple of examples. And different density of mechanoreceptors exists in different parts of the body. So you can see the homunculus I have on the right side there. This is just an image that tries to demonstrate where we are most sensitive, or where we have the most mechanoreceptors, most sensory receptors in terms of touch. So you can see that the parts of the body that has the most receptors and the most sensitivity are just represented as very large compared to other parts of the body. So we can see the hands, the lips, the tongue, these are all places that are very large on this image, so they're very sensitive. They have many receptors, whereas the torso is not very receptive. So it's very small on this image and it has fewer sensory receptors. So the next part of this lecture is pain. So the most important thing to remember is that pain is not just overstimulation of touch receptors. So we actually have specific pain receptors, and they're different from those that detect touch. Nociceptive pain is pain that's due to skin damage, and we have specific receptors for this as well, and we have those that are specialized for heat, cold, pressure, and chemical irritation. And then we also briefly talked about gate control theory. So this theory explains that there are inhibitory interneurons in the brainstem and spinal cord that block excitation. So they actually inhibit pain. So here again are a couple of terms for you guys to test yourself with. So now we're going to move on to lecture 5, which is perception. So let's talk about how we perceive, or how we have depth perception. So there are two types of cues that we discussed, monocular cues and binocular cues. So here's a list of monocular cues, and these are types of cues that rely on the use of one eye, and they're very useful to determine depth from 2D images. Now binocular cues actually require the use of both eyes, and we often use these cues to determine the depth in the 3D world. So for example, binocular disparity. So binocular disparity refers to the difference between the images coming from each eye. So the images are slightly different, or slightly askew from each other, and we can use this information to actually determine how far away the object is. And we can actually use this to determine how far away things are when they're quite far away. And that's contrary to convergence. Convergence is the actual inward turning of our eyes, so our eyes turning towards each other. And we can use this to determine distance of things that are close to us, but not very good at determining distance of things that are far away from us. So now I'm going to move on and talk about the 4 laws of grouping for Gestalt psychology. So the first one is law of similarity. So this just suggests that we like to group things, or we will frequently group things based on similarity. So if you were to look at these two pictures I have here, you might say, oh, well, I see 3 lines of black circles and 3 lines of white circles. You're not likely to say I see X number of circles and some of them are black and some of them are white. And then the next one is law of proximity. So here you're likely to say, oh, I see 3 groups of little white and blue circles that are of a particular size, and then I see 1 large group of these circles. You're not likely to say, oh, I see, the whole number of shapes that you have there. We're more likely to group them based on which ones are close to each other. And then we have law of closure. So this suggests that we'll actually fill in the gaps to make particular shapes. So if you look at the sort of square and circle that I have there, you're not likely to look at those 2 figures and say, okay, well, I see a series of horizontal and vertical lines. Some lines are placed at an angle. You're more likely to fill in those gaps and actually say, okay, this is a square and there's some pieces missing out of it. And then I have a real life example, so the actual IBM logo. So we can clearly read that as IBM, but it's actually just made up of a series of white and blue lines. And it's the fact that we can actually close in those lines to come up with a shape, and that we automatically do this, that makes it an effective logo we can actually read. So then we have law of continuity, also called law of good continuation. So this just suggests that we'll perceive a particular image as having the best continuity that it could have. So if we're looking at those 3 blue wiggly lines that I have down at the bottom there, we're more likely to perceive it as the one that's in the middle, so one long squiggly line with a shorter line crossing it, than we are to perceive it as one that's to the right of that, which is sort of 2 V-shaped lines that are opposite of each other. And again, here are some terms that I have for you guys for Lecture 5. So now we're going to move into Lecture 6, which is about consciousness. So consciousness involves being aware of our environment and ourselves, so paying attention to things in our world. So I have a couple of terms here for you as well. Selective attention. So this is the ability to focus your attention on one thing and ignore other things that are going on around you. So the classic example of this is the dichotic listening task. So this is the study where researchers play 2 different messages, one to each ear of a pair of headphones. And then the participant wears the headphones and they're told to pay attention to either the sounds or the story coming from the left ear or the story coming from the right ear. So what they found out is that even though the stimuli were still entering each ear, the participants were only able to recall information from the ear that they were attending to. So they had selective attention towards that one particular stimulus. Then we also talked about inattentional blindness. So this is when we fail to notice unexpected objects in our surroundings. So we're paying attention to something so closely that we miss something else. So for example, the video where you are asked to count how many times a basketball is thrown around a circle, and then at the end of the video you're asked, well, did you notice the monkey or the person in the monkey suit? And no one's really seen it because they've been focusing on counting the number of passes of the ball. So the next thing you want to know about is how do we quantify consciousness? So there are a couple of different ways we can do this. One is behavioral measures. So for example, the Glasgow Coma Scale that is discussed in your book, where we use different types of behavioral activity to actually give a score as to how conscious a person is, or where on a continuum of consciousness a person can be positioned. We can also use physiology. For example, we can use tools like EEG and fMRI to actually take measurements of what's going on in a person's brain, and we can come to a conclusion based on the results of these types of tests, what level of consciousness we think they have. You also talked about circadian rhythms. So there's a couple of diagrams in your slides. This is just one of them. So you want to recall that circadian rhythms are those natural rhythms that occur within your body. And at the very end of your consciousness lecture, you did talk about the stages of sleep as sort of a lead up into the next lecture. So you want to know the different stages of sleep, and then the waves that are associated with those. So I have a diagram here that represents that as well. And finally, here are some terms for your lecture 6. So now we're going to move into lecture 7, which is about sleep, our second last lecture. So why do we sleep? There are a couple of theories. So we have evolutionary theory. So just recall that this suggests that inactivity at night promotes our survival as a species. And then restoration theory. So sleep recharges our bodies, and this is critical of our physical health. There's also memory consolidation theory. So memories are strengthened during sleep. Here are a couple of sleep disorders that we talked about and that are also mentioned in your book. So we have insomnia. So this can be a couple of things. It's most likely chronic inability to either fall asleep initially, to maintain sleep, so stay asleep throughout the night, or to experience a restful sleep. So having insomnia doesn't necessarily mean that you can't sleep ever at all. It just means, it could mean that you just aren't able to experience sleep that you actually find helpful or restful. So even though you did sleep, you wake up still feeling exhausted. The next one we have here is narcolepsy. So this is the inability to stay awake or control our wakefulness. And what actually happens here, the underlying cause, is that your RAM, your rapid eye movement sleep, actually invades your waking unconscious. Then we have REM sleep behavior disorder. So this actually occurs when this paralysis that occurs during REM sleep of your body is inhibited. Then we have sleep apnea. So this is irregular breathing during sleep, so it's quite common, and it's actually caused by sagging tissue that happens when your sleep induces relaxation of these tissues. You also touched on some theories about dreaming, and these are covered in your book as well as in your lecture slides. Psychoanalytic theory was one of them, also memory consolidation theory and activation synthesis theory. And here are some of your terms for lecture 7. So the last lecture that I'm going to go over is lecture 8, which was on drugs. So I have a couple of things here, agonists and antagonists first of all. So agonists are drugs that increase the effect or the effectiveness of a particular neurotransmitter. So it doesn't necessarily increase how much neurotransmitter is within the synapse or within the bloodstream, but it does either increase it or it will increase its effectiveness. So for example, we could block the reuptake of the drug. So block the reuptake of the neurotransmitter, by the drug. Once the neurotransmitter is released into the synapse, everything that's not used is taken back up into the presynaptic cell. But if we prevent that from happening, it stays in the synapse and it's more likely to be taken in or attached to a receptor of that postsynaptic cell. We could also have drugs that actually just mimic the neurotransmitter, so they'll just go ahead and bind to those postsynaptic receptors and actually have the effect that the drug would have. And this is another way for a drug to be an agonist. Then on the other side, we have antagonists. So these are drugs that reduce the effect or prevent the effect of the neurotransmitter. And there are different ways of doing this. One is actually reducing how much neurotransmitter there is, and another way is by actually blocking the receptor for that neurotransmitter. So in an example, with an agonist, it would attach and then have the effect of the drug. With an antagonist, it would attach but then have no effect or a different effect, and it would actually block that receptor so that the actual neurotransmitter could not attach to it. So here I have just a diagram of the different types of drugs, or the different categories of drugs that you went over. So first we have depressants, which reduce your activity. We have opiates and alcohol as examples here. So opiates again, codeine, heroin, over-the-counter painkillers, anything that's derived from opium. And then we also have alcohol, which is also a depressant. In your book, we also talk about sedatives as being depressants. Then we have stimulants. These increase your nervous system activity, and people often take them for euphoria and for alertness. So for example, MDMA, which is ecstasy, or amphetamines. There's also cocaine, and of course caffeine and nicotine are stimulants as well. And then finally we have hallucinogens. So these are drugs that people refer to as being mind-altering. They can cause hallucinations or other effects, for example, LSD or acid. And then we also have marijuana, and marijuana is just given an asterisk here because some people also classify it as a depressant, and in some cases it's actually given its own category because it does behave a little bit differently than some of the other hallucinogens. So now I'm just going to go over tolerance versus withdrawal. So tolerance happens when there's a reduced responsivity to a drug, and this is in order for the body to maintain a homeostasis. So as someone's tolerance increases, the subject has to take more and more of that drug in order to get the effect that they want. So for example, somebody who drinks alcohol quite often, they will build up a tolerance and they'll have to drink a lot more alcohol to get the high or the sensation that they want compared to somebody who doesn't drink very often. Now withdrawal results from a compensatory response that are responses that are engaged to maintain a homeostasis. So this includes a constellation of symptoms that occur when a drug is ceased. So somebody who's taking a drug or has is having an effect of a drug and then that stops, they'll experience a certain constellation of symptoms that are referred to as withdrawal. So for example, when somebody withdraws from opiates, they can have flu-like symptoms. So somebody who's been taking opiates for a long time and they stop, they might get nausea or diarrhea or pain, fever, chills, other types of symptoms that are associated with the flu. And finally, here are some terms that you can study for Lecture 8. So again, a really good idea might just be to pause the lecture at each of these term slides and just sort of test yourself and think about if you know how these terms connect to each other and if you can discuss them in your own words. So like I said earlier, I hope that this was helpful for you. I'm also going to be uploading a lecture with some practice questions in it. So what I'll do is I'll keep the practice questions from Thursday's lecture or Thursday's review that will be helpful for you or that you will be tested on. And then I'll also add a couple of extra ones to sort of fill in the gaps where I'll be taking out some of the lecture or some of the questions that would not have been on material that you covered. So you can look forward to that being posted shortly. I'm hoping all this will be up and ready for you tomorrow morning, which is Monday morning, so that you will have the day to take a quick look at this lecture, help test yourself, and make sure that you really have everything nailed down. So thanks for listening, and good luck on your